Morning, everyone. Hopefully y'all can hear me. I see folks starting to join in. We're going to give a minute for people to get settled and get comfortable as we get going. We have all of our speakers here. I'm hoping you can see my screen. We'll be recording this as you joined. So if you want to have it saved for posterity later. I'm going to give a few more minutes because it's worth doing that as you get people on. So I see all the speakers and attendees. This is great. See folks starting to dial in. All right. I want to welcome everybody. I might as well get started. And, you can always stop as we go to our first in a series of the Common Fund Data Ecosystem public webinar series. And I'm really excited to be here. I'm part of the Knowledge Center. My name is Noël Burtt. You'll hear a little bit more about me and our team a little bit later. And I just want to get you all oriented to, webinars. Many of you join webinars, and I'm really thankful for you joining us on a Friday for a webinar. This is going to be a classic webinar, but we do want this to be interactive. You can pop your questions in the chat or a Q&A or raise your hand. We can unmute you and all that good stuff. And if it's anything urgent, Mackenzie's going to be monitoring the chat and the QA to make sure that we can address questions as they go so that we don't end up going too far ahead of you and you forget what you wanted to ask. So we're going to have many speakers today, and I want to start with a little bit of setting the tone for us all as we go through. So with that, I want to just sort of start with an image that I think really cleanly states our purpose and is symbolic of the Common Fund data ecosystem to me. And it's really about the fact that the reason you are all here today is because you're curious. And you're curious because you're scientists, right? And that's really the essence of what we all are doing as scientists. We are curious. And part of the reason why we're doing a public webinar series is because humans do science. And with all of our assumptions as humans, our context, our approaches, our expertise that we bring to interacting with the world, we observe it in different ways. And so our goal with these public webinars, particularly as the Knowledge Center, which you'll hear more about, is to engage with you and really understand you, because you power the discovery of all these scientific resources that we are so lucky as scientists to have at our disposal. So today you're going to hear about one purpose, which is the Common Fund data ecosystem, which brings together a set of resources and data repositories funded by the NIH, which George is going to speak a little bit more about in a moment, but makes them accessible to the world. But you as a user may not even know some of these exist. So our job with this public webinar and with the Knowledge Center and the other centers you're going to hear from over the course of the next year is to make that easier for you and bring that to you and actually begin a dialogue and a conversation with you as users as to in terms of what type of scientific questions could be asked with the data inside of these resources that we as generators and builders of these resources haven't even thought of yet. So this is really meant to be a conversation and a way to engage you as humans, both as users, contributors, and curious wanderers as we go through this webinar series. So today, your first foray into this will be an overview of the Common Fund data ecosystem from our colleagues at the NIH. And then you'll hear the first in a series of tours through the components of the data ecosystem. And we call these data coordinating centers, or DCCs. So you'll be hearing from the Metabolomics Workbench later today, really in the focus of what this resource is, but then how it fits in the broader ecosystem and how we might, as a community with you all, find new and exciting ways to analyze data from these various DCCs. And then the last part of our conversation today will be an initial glimpse as to how we hope to expose you and work directly with you, the wider scientific community, on what scientific questions and ideally discovery is possible within this system. And this is really, to me, the sort of a challenge in all of online science, if you will, these days. As a scientist, 15 years ago, we weren't all using lots of resources. We were maybe using one or two of them in our daily lives, but now they're everywhere. And I think sometimes it feels a little overwhelming to know what resources are out there, which ones to use, or yourself, you're using so many in your daily work that it feels a little bit like exhausting. We want to make that simpler with this webinar series and with the tools that the Common Fund Data Ecosystem is building. So with that, I'm going to stop and pull it over to our colleagues at the NIH. Actually, I think I'm running your slides, right? Right.
Yep.
So that's great. That makes it easier. And I will switch sides. Like, George, would you like to introduce yourself while I get your slides up and running?
Good afternoon. I'm George Papanicolaou, the program leader for the Common Fund Data Ecosystem, or CFDE. In our presentation, I'll provide some background of NIH's Common Fund, managed by the Office of Scientific Coordination and the Office of the Director, and then a description of Phase 1 of CFDE. Following that, Christy will be talking about Phase 2, which we are currently implementing and where to find our funding opportunities. Next slide. Next slide, please. There we go. Thank you. This slide outlines the features of Common Fund programs and the types of activities pursued by our programs to catalyze biomedical discovery. Some important features of Common Fund programs include substantial investments ranging from tens of to hundreds of millions of dollars. They are designed to achieve a series of ambitious goals within 10 years, and they are intended to have a significant impact on an area of biomedical research. Our programs do the following in ways that no other entity is likely to or able to do. One is accelerating emerging science. For example, our Human Microbiome Project expanded the field of microbiome research by leveraging recent advances in sequencing to characterize the microbiomes of healthy adults on a scale never before possible. Two, enhance the biomedical research workforce. For example, our Diversity Program Consortium is testing new ways of recruiting and retaining diverse populations into the biomedical research workforce. Next, remove research roadblocks. For example, our transformative cryo-electron microscopy program is making state-of-the-art cryo-EM equipment and training available to the research community to broaden the use of this game-changing technology. And then, finally, support high-risk, high-reward science. For example, our HRHR research program supports exceptionally creative scientists pursuing highly innovative research with the potential for broad impact in biomedical, behavioral, or social sciences within the NIH mission. This support includes 2 initiatives for early career investigators. Next slide, please. Thank you. There are certain criteria that need to be met to ensure that the programs are suitable for Common Fund support. These programs should have a high impact and create new research or clinical care approaches. Next, the program must have a defined goal and deliverables that can be achieved within 5 to 10 years. The output of these programs should enable the mission of the multiple ICs, or institutes and centers, and be cross-cutting, addressing complex issues that require trans-NIH teams to design and manage. There must be a reason why strategic coordination is required. And finally, these programs should provide new solutions to specific challenges and not be something that another entity would be able or likely to support. If there are similar efforts, the Common Fund program should be tightly coordinated to prevent duplication of effort. Next slide. The NIH Common Fund is a collaborative endeavor and requires input and effort by many members of the biomedical research and NIH communities. During strategic planning, we gather diverse input from NIH leadership, staff, and the broad biomedical research community. This includes identifying potential new Common Fund programs and shaping and refining new program ideas once they are selected. We assemble consortia of multidisciplinary, innovative researchers collaborating to tackle a shared ambitious goal. The time-limited and goal-directed nature of our programs makes a collective effort and diverse expertise necessary for success. We manage our programs in partnership with you. Each Common Fund program is run by trans-NIH working groups with leadership provided by a few ICs. Directors from these ICs or senior staff serve as program co-chairs, providing leadership and guidance for our programs. These directors nominate members of their staff to serve on programs within their working groups. For example, this shows how we work across NIH. Dental was a lead IC for the Human Microbiome Project and works with us to administer some of our high-risk, high-reward research programs. NIAMS is a lead IC for MotorPack, which is one of our Common Fund programs. Next slide. Here we see how Common Fund programs are aligned within 3 general categories, reflecting their mission and expected outcomes. On the top left, we have our transformational science and discovery programs. These programs are designed to establish new scientific principles, models, and research resources that will transform scientific knowledge and pathways to discovery. Examples include 4DN, XRNA, and MotorPack. On the top right, we have Reengineering the Research Enterprise programs that are designed to transform how we do biomedical research and behavioral research, how we make the biomedical workforce as robust as possible to ensure new perspectives and ideas contribute to discovery, how we transition that research into prevention and therapies, and finally, how those successful prevention and therapies can be shared broadly. Examples include COMPASS and the Undiagnosed Diseases Network. Last but not least, we have catalytic data resource programs. These programs are designed to manage and develop data for scientific discoveries by accelerating research through data resources. Examples include Bridge to AI, Kids First, and of course, my favorite, CFDE. Next slide. Next slide, please. So CFD's mission is to foster scientific discovery through the use and reuse of data and resources generated by Common Fund programs. We have 3 aims. Enable users to search multiple Common Fund data sets and use them together. Provide training and outreach to help people work with Common Fund data in the cloud, and to coordinate and integrate infrastructure and activities to create a cohesive ecosystem. To our right, you can see the elements of the ecosystem. Data consumers interact primarily in the top half, and data producers are represented in CFDE as data coordinating centers, or DCCs, for each Common Fund program on the bottom. Note within the ecosystem, individuals can be data consumers and producers. And through CFDE, we funded and enriched both halves through research, products, and data integration and discovery. One of the conditions of many of our awards is that generated products and harmonization and integration need to be shared with the greater scientific community, as well as parent Common Fund programs that generated the data. Next slide, please. So where is CFD today? So CFD has existed for approximately 4 years. As we're currently in the transition of phase 1 to phase 2, let me give a bit of background. During the pilot phase, CFD was organized into a hub and spoke model, which consisted of a single coordinating center and engaged Common Fund program DCCs that participated by submitting metadata to the dataset catalog, performing fairness assessments, and partnering to integrate data and create digital resources across Common Fund programs. To the right of the slide, you can see the hub and spoke model and the various Common Fund program DCCs that are currently part of CFDE. And DCCs have brought in data that include molecular associations with health and disease, cell scale anatomy atlases, and drug effects on cells and genes. An exciting part of CFDE has been supporting data reuse and trans-Common Fund integration through 42 research awards, which are classified as RO3s to enhance the utility and use of Common Fund datasets. We have a new round that you can see later via a QR code that Christy will present, and that represents all of our funding opportunities. Note that all this effort is only made possible due to the efforts of the various centers, the DCCs, and the entire Common Fund data ecosystem team, who you'll be meeting in future webinars. I'm pleased to hand over the presentation to Dr. Christy Kano, the program manager of CFDE, who will talk about the exciting changes in phase 2.
Great, thanks, George. Next slide. Yeah, one more, thanks. Okay, so I thought we would spend a moment just talking about kind of how we're going to be structured as we move into phase 2, which officially kicked off about 6 months ago in October. And then I'll talk a little bit about kind of what we're trying to do and what the goals are for phase 2, what we're trying to achieve. So the primary structural change as we move into phase 2 is that we're moving away from one centralized coordinating center to a group of 5 distinct but interconnected centers, each that has a sort of a unique domain. 4 of those 5 centers are kind of technical in nature, so I'll talk about each one in turn. And then I'll talk about the last one. So the first one is the Data Resource Center. So its job is really to set up sort of our program landing page, the place where you can go to learn about all things CFDE, house information about all of our participating Common Fund programs, and all the different activities that we have going on within the ecosystem. In addition to the sort of what we're calling the information portal, or like the landing page or framework for the program, they're also building a data resource portal where you'll be able to come in and query for data across Common Fund programs. They're also building out sort of a variety of tools and resources that will link to that data and provide you with resources to help use the data. The next center is the Knowledge Center, and they're similarly, but a little bit different. They're creating a portal that will allow you to go in and search for knowledge that's been generated by participating programs. So with both of these, the output is sort of a portal that allows you to come in and kind of search across participating programs. Hopefully we will have joining us very soon in the next 6 months or so 2 new centers, the first of which is the Training Center. So their task will be to coordinate training across the ecosystem. So they will not be necessarily developing or delivering all of our trainings, but they'll be coordinating all of the training activity and helping us fill in the gaps as we go. And the fourth center is the Cloud Workspace Implementation Center. Again, hoping to stand this one up pretty soon here in the next 6 months or so. And it will provide a cloud workspace where users can import data, Common Fund data, as well as other types of data, and analyze it in the cloud. The final center is the Integration and Coordination Center, and it's a little bit less technical. Its primary focus is really on ensuring that we have cohesion across the program as a whole. They're also tasked with responsibilities around evaluation and sustainability. The role of the DCCs that are participating will remain largely the same as we move into phase 2. That is, they'll continue to contribute their metadata, as well as help us build the tools and resources that will help make Common Fund data more interoperable and reusable. We do hope, though, and we expect that the number of participating programs will continue to increase in phase 2, and that it will include some new data types that aren't currently represented within the portal. Next slide. Okay, so how are we building the data ecosystem in phase 2? So an ecosystem is defined as a biological community of interacting organisms and a physical environment. So I think the most important part of any ecosystem is the people. So one of the things that we're really going to be focusing on phase 2 is raising awareness for CFDE among the community and the great resources that we have to provide that will help users make use of Common Fund data. In terms of the sort of physical environment, if you will, that's really the products that we're going to be building, and I'll talk about those in just a moment. And then last thing we're trying to do in phase 2 is really start to the extent that we're able, sort of systematize some of the processes that are involved in making sure that data can be shared and reused easily, even after Common Fund programs have ended. So I'll spend just a moment talking about each of these sort of 3 areas in turn. Next slide. Okay, people. So as I mentioned, one of the primary goals is to help make people aware of the different resources that we have that we're going to be providing for the community that will help them take advantage of the great data sets and resources that have been generated by Common Fund programs. So this is going to include things like community building events, like hackathons and workshops, conferences, presentations, etc. In addition to that, we also want to make sure that the community has the resources it needs to make effective use of those tools. So we'll be offering a lot of training and mentoring opportunities. We're hoping to provide a diverse array of trainings, so both in the format that we offer it, online and in person, as well as to like a diverse group of audience. So we really want to bring in a wide variety of users, from people who are maybe primarily bench scientists, don't have a lot of computational expertise, making sure they have the skills that they need to start to reuse some Common Fund data, as well as some trainings for more advanced users that maybe have a computational background so that they can take best advantage of the resources that the Common Fund has to offer. So those are kind of our 2 main emphases in terms of the people and the community in phase 2. Next slide. Okay, so in terms of products, I kind of think about the products that we're developing in sort of 2 main buckets, if you will. The first bucket of tools are tools that help make the data and the knowledge from Common Fund programs more findable, so helping to connect users with the data, resource, or knowledge that they need. So those include things like the CFDE portal, where you can go to learn about the program and the participating programs, the data research portal, where you can go in and search for data, as well as like tools that will help you make best use of that data. And then the knowledge portal, where you can go in and search for knowledge that's been generated by participating programs. So in addition to these tools that help make it more findable, we'll also have a variety of tools that are going to help harmonize disparate data sets and help users analyze the data in a meaningful way. Lastly, we're also, as I mentioned, hoping to develop a cloud workspace where users can import Common Fund data alongside their own data or data from any other program and do their analysis in the cloud. Next slide.
Christy, we also have a great question, which I think is timely. It's actually, I'll read it out loud. This is from Hunter. The current R03 funding mechanism requires integration of 2 CF repositories. That's a bit short-sighted. Why not require 2 or more repositories or knowledge bases, one of which is the CFDE data? I think that's an interesting question. What are your thoughts on that?
Yeah, I'm sorry. So is this the R03 program that you're talking about, or?
Yeah, that's what he was asking a question about.
Yeah, so I think it's a great idea. I absolutely think it's something we will consider in future rounds. I think, our primary objective is really to make sure that we're driving sort of novel reuse of Common Fund programs. But I think your point about, can and should we start to require, either more than 2 programs or the incorporation of, data sets and resources outside of Common Fund. That's a great point, and definitely something we'll be thinking about as we move forward.
Great.
Any other questions or?
And then George is going to type an answer to that. That's very good. And then Hunter has a great point, which is you need integration of CF without outside resources as well. Yeah,
no, absolutely. So I don't know if, one of the things we haven't had a chance to speak about just yet, but so we have what we call a partnership program, where it's so in the past it was 3 or more Common Fund programs coming together to submit a proposal about how they would like build a tool or a resource or harmonize data sets that pulled resources from three or more Common Fund programs. And one of the requirements we incorporated into our most recent funding announcement back in 2023 was a requirement to include a non-Common Fund group or collaborator. I don't think we went so far as to specify that you had to incorporate a non-Common Fund resource or data set, but the idea is that, we totally hear you that like at some point, for these things to be really useful, it's going to be helpful to integrate them with the sort of broader ecosystem of resources that exist outside of Common Fund. And so that was kind of our intent in making that requirement that you had to have a non-Common Fund collaborator as part of that partnership project. But I think your point is well made.
Yeah, and I'll just echo that, Hunter, one of the things you'll hear hopefully later is at the Knowledge Center, one of the things we're really hoping to do is figure out and work with people just like you who have ideas on what we could incorporate, because some of these analyses are not super costly, right? Because they already exist, and with a little bit of effort, you don't need a huge, a year of R01, R03 funding. You can actually do it with, a little intrepid investigators like yourself who are interested in doing an analysis, and then we could immediately make those accessible in the Knowledge Center. So that's something that we really hope to achieve over the course of the next 5 years with the research community. So with that, I'll turn it back to Christy.
Yeah, great. Thanks. Great questions and great feedback. So thank you. Okay, so last thing is process. So, during the pilot phase, we did some exploration around sort of, thinking about sort of life cycles. But I think as we move into phase 2, we're really trying to the extent that we're able, systematize some of the processes that are important for making data sharing sort of easy and sustainable. So, we'll continue our work around developing terminologies, ontologies, controlled vocabularies that make it easy to share data. Or easier to share data, as well as thinking about, how can we, are there ways that we can, best practices or sort of a life cycle process that we can adopt that makes sustainability easier. So starting to think about sustainability earlier in the life cycle and maybe having sort of a series of checkpoints as, as Common Fund programs execute, so that when we get to the end of the life cycle, it's not, we have a good plan in place. I think people in general are sort of thinking about sustainability right at the very beginning when they, when they're submitting their applications, and then right at the very end when the project is ending. And so what we're really hoping to do is think about it as a life cycle and develop a process so that when we do come to the end of a life cycle, we have sort of a plan in place that's relatively easy to execute to ensure that the data and resources generated by the program are sustainable. Next slide. Okay, so this is the slide that George mentioned where you can check out our current funding opportunities. So, check back often. We're posting new things all the time. So really encourage you to take a look at them because, we would love to have your help in building the ecosystem and participating in the ecosystem. So that is where you should go. Next slide. Okay, I think that's it for us. Thank you. George and I are happy to take any other questions that you might have.
Awesome. Thank you, George. Thank you, Christy. Any questions? I'm going to turn it over and get Mano and Shankar up and running. I'm really excited. We're running probably a little bit behind time, so I'm going to keep it to 15 minutes if you guys can. But so this is your first tour and first exposure of one of our DCCs. And what's great is the reason we wanted to use this one as our example is we've really used Metabolomics Workbench as an example for the Knowledge Center also of how do we make some of the results accessible to someone who may not even know what metabolomics is and may want to use it in the research. So you can see this is a theme that's going to come through the rest of the session. So with that, I will turn it over to Mano and Shankar to take the com.
Thank you, Noël. Hello, everyone. We are very happy to provide a very, very quick hitchhiker's guide into the Metabolomics Workbench. This journey started about 20 years ago when we got a large grant called the Lipid Maps, it's a GLUE grant. And at that point in time, the last time when anything with nomenclature or organization of metabolites, especially lipids, was done was in 1960s. So it was kind of, all over the place, and mass spec was just coming into picture. And then came the next 10 years, which really mushroomed into large measurements of metabolites, going from hundreds at a time to tens of thousands of metabolites. And this launched the initial project. NIH, recognizing the importance of metabolites, started in the early 2000s. A project, a common fund project, which involved the National Metabolomics Data Repository and the Metabolomics Workbench, along with funding 10 other groups for generating data, vast amounts of data. And this journey now is continuing with funding from Metabolomics Workbench from the Common Funds, Data Commons Workbench from the NIH CFDE Phase 1 project, and the CFDE Data Resource Center. I wear two hats. I wear the hat of the co-PI along with Dr. Avi Mayan for the Data Resource Center, and I've been the PI for the Metabolomics Workbench for 12 years at this point in time. So a lot of people have contributed over a period of time. I'm not going to acknowledge every one of them. I'll just going to only introduce Mano, who will then talk about the next 15 minutes about Metabolomics Workbench. Let me walk you through why metabolomics in a few seconds. We know the central paradigm in biology. DNA gives RNA gives proteins, but then proteins act on metabolites. Metabolites are ubiquitous in human physiology and in mammalian physiology and other physiology. And these are functional endpoints of physiology and pathophysiology. They are kind of reasonable scale of analytical challenge because they are small molecules to medium-sized molecules. They are measurable from serum and blood, which is fantastically important because they're easily accessible body fluids. And then they are a direct mirror of environmental influences, malnutrition, like diet, exercise, medication. And so it really is an endpoint. And we can quantitatively metabotype individuals, just like genotyping. We can characterize the dynamic state of an individual through metabotyping. What are the challenges in metabolomics? Well, how do you measure metabolites? Today, mass spec can produce tens of thousands of metabolites in one single measurement. How quantitative is metabolomics? With standards now, it's become extremely quantitative. So we can longitudinally monitor a human being for their metabolites in blood or serum, and we can look at the changes. How can we analyze the omics measurements to obtain metabolite information? We have been developing tools the community has been developing tools for the last 2 decades towards mining these metabolomics measurements. How can we organize, store, and use metabolomics data? I would like to say that National Metabolomics Data Repository, which is NMDR, and the EBI metabolites are the two unique resources in the world which essentially provide organized, store, and provide metabolomics data. How can metabolomics data be integrated to provide a systems view of biology? This is the quest of jointly Data Resource Center, Knowledge Center, as you'll hear later on, and of course, Metabolomics Workbench. What are the implications for systems medicine? Can we metabotype an individual? Please wait for the future. This is something we are actively engaged in terms of thinking about a stamp, rubber stamp of a dynamic state of a human being. What NIH decided to introduce this in the Common Fund program. They wanted training in metabolomics. They wanted comprehensive metabolomics resource course, metabolomics technology development, data sharing and international collaboration, and reference and synthesis standards. This is what Metabolomics Workbench has led to. I'm going to now turn it over to my colleague, Dr. Mauro, who will go through the next, 10 minutes, the next few slides. Mano, over to you.
Thank you, Shankar. So I'm going to provide some details about what the Metabolomics Workbench resources are. So starting with this slide, here we get a glimpse of different assets that we have in Metabolomics Workbench. For example, we have over more than 3,000 studies deposited, out of which more than 2,700 have been made public. Similarly, many of them, more than 1,000, are related to human studies, and we have more than 3 lakhs samples, and we have some other details provided here, which I'm not going to cover in complete detail. So let's go to next slide. Next slide, please. So this slide provides an overview of how, starting with the data and metadata deposition into the system, how we channel it through different processing steps, and then to various tools, and how it is visualized on the Metabolomics Workbench website. So starting with the experimental data that is generated by different data metabolomics researchers, we have an online submission system where we have the ability to provide named metabolites, which and all that information, along with the other metadata information, is deposited into a relational database. Next, please. So once this data and metadata is in the database, we have a series of tools as part of a comprehensive toolbox, which can be applied to these different studies, and we can do different types of analysis, some of which I will cover a little bit later. In the case of untargeted data, the metadata part goes into the regular database that we have, and the detailed untargeted data goes into a different sort of file-based system. Next, please. Because it's untargeted data, in this case, M over Z values are not being able to map to a particular metabolite, so it goes through a different pipeline, and eventually it is also channelized to a different set of tools. And again, some of that data can be visualized in various means. Next slide. So this slide provides an overview of how the data deposited into NMDR has grown over time. As we can see here, it has continuously been growing, and particularly in December 2015, an automated online deposition system was introduced based on all the experiences that we gathered over the previous 3 years, and we saw a substantial increase in the number of studies deposited after that, and the momentum has continued. Next slide, please. So in Metabolomics Workbench, in different sections, we can look at the data based on different aspects. For example, we can browse the studies based on the species name, for example, whether it is from human, mouse, or rat, and we can see those 3 are the top represented species across all the studies. Similarly, we can look at it based on the disease, for example, cancer and diabetes, and then obesity. Those are 3 are the top three diseases that we have seen in the samples in the studies deposited. Next slide. Similarly, there are other ways to sort of look at, slice the data and then see them and further follow through. For example, based on the sample type or the type of human pathways that are covered by the metabolites that have been measured in these studies that we can see on the right-hand side. Next slide, please. So I will briefly go over the capabilities of Metabolomics Workbench. For example, we can submit metabolomics data and then query and search across the studies based on certain keywords corresponding to different types of attributes, and then be able to download the data for further custom analysis. And then, of course, use various tools on the website itself, for example, for visualization, analyzing the data, and other types of integrative analysis. Next slide, please. So before I go into the actual examples of studies and analysis, different analysis tools, we wanted to point that we store much of the data, much of the metadata in a format called MW tab, which essentially corresponds to Metabolomics Workbench tabular format, which has various sections such as metadata section, data section, and some additional information can be provided as comment. And a view of that is provided, a file view of that arrangement is provided on the right-hand side. In the metadata section, we have information such as the project, study, experimental variables, referred to as factors or different parameters that define a specific condition under which that, for which that sample corresponds to, and other related information. In the data, we have the actual data values and tables. Next slide, please. So now going to the actual different parts of the website, the most prominent one from a user perspective is to browsing and searching through the different studies, where we can browse them and search based on different aspects, and also use REST services to fetch the data from data and metadata from Metabolomics Workbench. Next, please. So, for example, when searching for the studies, this table provides a overview where we have the ability to sort them based on different columns like study ID or study title or species or analysis and so on. And then we can select a particular study and follow on that further. In this table, we also directly provide a way to download much of the data related to that study. Next slide, please. Besides MW tab, another very important feature of RMDR is that we use a reference metabolite system called as RefMet, which provides harmonization and standardization of metabolite names. And that's very important for being able to integrate the data from different studies or across other resources. It's a unifying nomenclature. It provides the ability to perform comparative analysis across different studies. Next, please. In reference system, for many of the metabolites, exact structure has been also linked to a metabolite structure database. And then a comprehensive classification system is provided based on various labels of details, which is used for data visualization and statistical analysis. And all this facilitates integration with biochemical pathway tools that helps us map these metabolites to different further interpretation and annotation of these metabolites. Next slide, please. So going to various tools that are available for the study, we can select a particular study, and within a study, there could be different measurement modes, for example, GCMS, positive ion mode, negative ion mode, and so on. And once we choose that, we have a set of tools listed on the respective pages where we can click choose any one of those and then select the parameters related to those and then carry out the analysis. What is shown on the right-hand side is just a summary view of the results from different types of analysis, which some of which we are going to look at with respect to specific studies. For example, looking at study number ST001140, which is related to changes in the canine plasma lipidome after short and long-term excess glucocorticoid exposure. Okay, so what is shown here as a summary view is different types of analysis. For example, Kano plot analysis that we see on the right-hand side, the left bottom panel, and then we can also do class enrichment analysis as shown on the right-hand side. Next slide. Maybe I can pause while we address the questions.
Yeah, there's two questions in the chat. One about cumulative statistics. I think the question is, what is the URL for this cumulative statistic page, or what are the instructions for selecting it from the Metabolomics Workbench homepage?
So I can address that. We have not we have not shared that URL, but because largely that has been for internal use to gather that statistics. But I believe we should be able to provide that link. I believe the part of the reason it was not provided directly on the website is that in order to compute those tables, it requires a little bit of computation. I mean, searching through all the databases. But we can certainly look into, as long as it doesn't put too much pressure on our infrastructure, it should not be a problem.
Right. All right. I think we're good. Keep on going.
All right. There was another question. Yeah, certainly we can update that.
You can access it without logging in.
Oh, okay. Thank you. Okay. So looking at some of the tools for analyzing this data for study 1140, for example, we can do clustered correlation analysis, which we discuss on the next slide. So here we have some parameters, for example, what is the cutoff we want to choose for correlation, and which MS analysis we want to include in our study. Or I believe we also have the option to include all of them to get all the metabolites included. So what this shows is a correlation heat map for that data set, which kind of provides an idea which metabolite groups are more correlated to each other and so on, which we can further analyze. Another example is network analysis on correlated metabolites corresponding to mapping it to classification and also mapping it to fold changes. Next slide, please. So this slide essentially shows a tool provided by a Michigan group. And actually, this also kind of shows that we have the ability to, at least to some extent, incorporate compatible tools developed by other groups into the workbench. In this case, given the data matrix for metabolomics data, we have the ability to generate networks based on what is known as a debiased sparse partial correlation. And we can look at this network further based on various filtering and so on. Here is another example of building network based on a mixomic package. This one is non-interactive, but I believe we do have the ability to select different cutoffs for the correlation so that we can get more dense or sparser network here. And for all these results, we provide a way to download the results, whether it's a PDF file or in some other format. Then we have an extensive set of tools, or extensive set of means to extract the data from Metabolomics Workbench via the REST services. And this slide provides just a little bit a glimpse into that. The actual web page provides a lot more descriptions of how to construct the REST URL, including the example given here. Next slide, please.
Mano, we can wrap up quickly in the next 3 minutes, maybe.
Right, yeah. So besides the core Metabolomics Workbench resources, as part of the CFDE effort, we have been developing some cool tools. For example, what is shown here is called MetGene, which provides a workflow where starting with a gene or a set of genes, one can find what are the reactions and metabolites associated with those genes. And in Metabolomics Workbench, what are the studies where those metabolites have been measured and related information. Next slide, please. Another tool that we have developed is called MetNP, which is another workflow which lets one either analyze the data from one of the studies that Metabolomics Workbench, or the user can upload their own data in a compatible format, and we find out what are the differentially altered metabolite between two conditions, what are the pathways or classes to which they map, and it generates a network for the metabolites and the different pathways and various other results. Many of these tools are actually available through as Jupyter notebooks as well as shown here on our GitHub page link. Next slide, please. So now I think, Shankar, you can take over.
Thank you, Mano. So I'll wrap up in just 1 minute. Beyond metabolomics, we want to really derive biological knowledge in deep ways. And so this is already, Christy showed you the slide. I mean, Common Fund Data Ecosystem entered phase 2 with introducing DRC, Data Resource Center, Knowledge Center, Quick CW Cloud Implementation Center, and Center for Training, and the Coordination Center linking all of those. And so we are now entering into a phase where we want to marry the DCC in an integral way with everything else that is going on in CFDE. In fact, we have this diagram which was shown before, and we also provide, in response to Hunter's earlier question, we really provide a link to external repositories and resources such as, All of Us, Steady, TopMed, EPIC, breast cancer projects, and dbGaP, etc. And so, in fact, this is one of our goals in DRC to really bring in synthesis of all of these in a queryable way. And the biomedical research community, I mentioned to you about the Knowledge Center. The Knowledge Center is the key towards the next phase of building knowledge from all of these collective resources. And I will turn it over now to Noël to continue with the presentation.
Awesome. Thank you, Mano. Thank you, Shankar. That was great. We did pack in a lot today. This is our dress rehearsal for our first webinar. So bear with us as we throw a lot at you. Hopefully that's exciting, right? You're curious, so you're in, right? So I'm gonna take the calm for a little bit and give you this initial glimpse that we promised you of the Knowledge Center, what we're trying to do to translate a lot of what you heard today through the DCC for the Metabolomics Workbench and from Christy and George, and build a resource to some sort of being a jumping off point for the entire ecosystem. That's really what we're hoping to do. And I'm going to kind of cut my time a little short because I really want you to hear from Gaulton, who's one of the other MPIs on the Knowledge Center, but also is, to me, one of the ideal users for the Knowledge Center, because he is an investigator all by himself who thinks about, scientific questions related to metabolism and diabetes. And he works in the functional genomics space. And he is, to me, a perfect user because he may not have known what all these resources were. But as we started working on this project, it was really cool because as he sat in the room, he did exactly what I'm hoping that we can do with you all on these calls is go, ooh, what if we did this analysis? He's going to tell you a little bit about that in the end. So to begin, the Knowledge Center. Our initial plan was to bring scientifically valid results to the wider research community. We had 5 aims to do that. Well, to build a first blush of that, it's a lot to tackle, right? There's a lot of resources. Even just listening to Mano and Shankar talk about the richness of Metabolomics Workbench, it's a lot. They have an entire community, entire set of investigators, data, approaches that they house and really are the shepherds of. How do we as a Knowledge Center shepherd that to a broader community? It's a bit of a challenge, but here's how we decided to approach it. So I don't know if you know, many of us that are on the Knowledge Center build scientific resources also for human genetics. And this is an important example, I thought, because it kind of sets the tone for how we approach the Knowledge Center. For example, if you are a human geneticist and you have a favorite phenotype and you want to generate a list of genes to investigate, in a world where you don't have all these resources, you might go to one resource, the GWAS catalog, download all the summary statistics, run a meta-analysis in R, knowing all the proper scripts and processes and tools and steps you want to do. And then you're going to incorporate a bunch of other resources. You're going to go to this thing called FUMA GWAS. You're going to do literature searches. You're going to go to model organism databases. You're going to use different resources that provide gene expression results. And then finally, at the end, you may have a list of genes to investigate. You'll do this well. You know what you're doing. You're an expert. But sometimes you'd like it done for you and a little bit easier so that if you want it represented in one place, very simple, you can have it access to you. That's what we build with our human genetics resources. We don't take away all those details in between. In fact, we hope that you would want to go back and do those to sort of verify, but we make it accessible. And really what we try and do is we catalyze research using the 80-20 rule. But for those of you who like food and live in New England, here's the example I like to give. It's called the lazy man's lobster. If you love lobster and you normally eat lobster, you realize it's a messy task. You requires the bib and tools and butter, and it's messy, and there's a lot of things to do, and then you finally get to eat. But they have this thing called the lazy man's lobster, where the beautiful, elegant cooks behind you, they prepare this beautiful plate of butter lobster served directly for you, which sometimes suffices, even if you're an expert and you know exactly how to deal with the lobster. So I want you to keep that in mind when you think about how we built the Knowledge Center. We want to make so they can be readily accessible to you, even if you're an expert in that data type or in that scientific question. But if you want to dig deeper, you can, but you can get the results right to you. through the Knowledge Center. So how do we approach this? Well, we started with listening, doing research, and really collaborating with all of these data ecosystem members in the DCCs over the last 6 months. And we realized, wow, there are a rich host of data resources and tools. It's incredible. We have a lot of learning to do. And you as a user must have a lot if you actually wanted to take on the task of learning these. So how can we make this a little more simple for you? Well, what we learned is, even in the ecosystem, as investigators inside the ecosystem, we all wanted to put the information about all of these DCCs, what they do, in one place, so you can find them. We want to make those results and data more meaningful to an end user. We also want to enable things like more of the data being used, because some of the resources said only 1% of our data is used by 90% of our resources. We want to contextualize all of these DCCs in human disease context or in basic biology. We want to make sure they're sustainable, and we really want to use the resource to say what can be done with all of these data resource centers. So we attempted in the last 5 months, we're very new to this effort, and we're very excited to be part of it. We attempted to get water flowing through the system. I know that sounds silly, but that was really the goal. And we really want to make it accessible. We want to be vulnerable. In many cases, a lot of times people go off and they build resources and they build this beautiful, elegant system, and they're not sort of dipping their toes in the water to the researchers. And then it may not be exactly what you need. And if it's not exactly what you need, you may never come back. So our approach is really, we want to do this frequently and often and share with you what we've built and see if it's right. Tweak it, erase the whiteboard, start over. So what you're going to see today is our first pass at presenting the ecosystem in an interactive way, allowing you to navigate through the DCCs and represent results from a very few scientific vignettes that we've come up with together in a context of, say, a drug developer, an experimental biologist, or just the average person who's curious. So with this initial offering, we have all the information of all the DCCs curated in one place. We make it easy to get to. We provide like a jumping off point, a start, like on the cliff, right? And we hope this resource will reach a broader research context. So how do we do this? Well, we want to organize knowledge in 4 different categories. Without going into these in the interest of time, I'm going to basically more explicitly describe them. One is we want to present results if they've been generated as is by a specific DCC. This is, they've done the analysis, they've done the results. It's very simple. We want to make that accessible rather than having you go to that specific DCC. We also, in the case of the Metabolomics Workbench, as you heard today, a lot of the stuff you can do in Metabolomics Workbench, but you need to know what you're doing, right? You need to be an expert in metabolomics. What if we could pre-compute some of those for you in the context of human disease or your favorite metabolite? That's another vignette we'll tell you. What if we could generate results that the DCC itself would love to be doing, but may have published in a separate publication, has the data sitting on their DCC. All we need to do is run that method and present it to you. That's an example of GTEx you're going to hear about. And then finally, the most complicated one, which gets to, I think, Hunter's point a little earlier, which was perform a new analysis that's sparked by a scientific question. So take data from a resource, run an external method that creates new results from that, then add an external resource like genetic associations from the largest repository for genetic associations out there, and present results in the context of biology for the first time. So that's what you're going to hear about. That's what we've built. We're just beginning, and it's not perfect, but we want your feedback. So that's the whole thing is contextualized by human questions. And so that's what we're hoping is, if you have questions, if like from this webinar, one person is like, I would love it if they could insert blank. Run this analysis and produce these results from this DCC or this set of DCC resources. We'd love that. And that's what we want to do more of. The team that built all this is my fellow MPIs and this wonderful team. We built it in 6 months, which is pretty awesome. And it's just the beginning. So with that, I'm going to give you a little tour, and then I'm going to hand it over to Kyle to give a more specific tour. If you want to log in, it's CFDE Remember, it's new. We want feedback, but we want you to take a look at it. It's meant to get us all organized, give us a gateway to spark your curiosity, and contextualize your research. I'm going to look at the time, make sure, oh, I have 6 minutes. Holy moly. So I'm going to first start with just giving you a view of the landscape of the page, and then I'm going to quickly turn it over to Kyle to make sure that we get some time. So here's the page if you land on it. The idea is to give you sort of a glimpse of the ecosystem from the perspective of areas of biology or research. So the idea is if this page is interactive, it's meant to be sort of a navigational tool, but also sort of an exploratory thing to learn about the components of the ecosystem. So if you click on each of these, you'll see these dotted lines that connect to the DCCs. And they're thick and thin based on how relevant they are to that sort of context of study or research. This is not a perfect science, but this is our first attempt to sort of put this into a framework for you all who may not be familiar with what MotorPack is, for example. So if you hover over them, you'll see them change and whatnot. Then you can set your context and your entire experience can come after that. What we do then is if you set your context and you hover over each of these DCCs, you get a description of what these DCCs do in the context of that particular research frame. What's also nice is if you'll see the orange highlighting below, you'll see for that DCC, the omics data sets they have, and then how they organize their data, whether it be by different entities, whether it be by gene, variant, protein. And this is sort of how we imagine you as a researcher navigating through the rest of the resource, right? Because these DCCs span things that have in common, but they also do it in their own way. So you can choose to navigate the resource that way, but you can also choose to learn this as a learning opportunity. I'm going to turn it over to Kyle real quickly and not do 2 gene searches because I think Kyle will be best to do that. But if you want to learn anything about everything we've done so far, you have access to all of our webinars, description of the research context that we've done, so how we define those, documentation, how we've done each of these analyses, and then information on each DCC. This is what we meant by everything in one place. This is a lot, but it's also a start. It's the idea that each DCC is contextualized by the same things I told you on the front, and you can navigate and pull and play based on those. But you can learn about the foundational, primary, and secondary, and even tertiary knowledge generated in those resources, but also what's available in the KC. So with that, I'm going to turn it over to you, Kyle, to give a couple stories, particularly around Metabolomics Workbench and 4DN, that will be most relevant to this audience in the last couple minutes. That's okay.
Sure. Okay, I'll try to fit that into 2 minutes.
Sorry.
Okay, so I'm actually just going to talk about the Metabolomics Workbench results very quickly. So based on what Shankar and his group talked about, they have collected metabolomics data and specifically provide the results of studies that have compared metabolite profiles in various conditions. And one thing that we thought would be valuable to collect for the Knowledge Center was a meta-analysis of associations of metabolite levels for the same trait or disease across this resource and provide that on the trait page on the Knowledge Center. So briefly, what questions can we ask from metabolite data of case control samples? So this may differ depending on the type of user you are, but if you're in pharma, drug development, for example, what metabolites might represent biomarkers of a disease? In other contexts, can these metabolites give us insights into the underlying mechanisms of disease? If you, for example, go to the type 2 diabetes page on the Knowledge Center and look up metabolite associations meta-analyzed across the data in the workbench, this will show you a plot of the associations ranked by p-value. And if we look at the top associations for type 2 diabetes, there are some that make sense, like glucose, for example, as some of the top results for type 2 diabetes, and the URL is here, but you can also go search on the Knowledge Center for type 2 diabetes, and this is one of the modules that you can see results for. So can this give us novel insight into metabolites that are associated with disease? So in several cases, I just pulled out a couple of examples of where these associations may provide useful information for understanding metabolite levels in disease. So for Alzheimer's disease, for example, glycine was one of the top associations with Alzheimer's disease compared to controls. And glycine has had some evidence, although not definitively, for a potential role in Alzheimer's disease. Another example that I thought was interesting, Mauro studies type 1 diabetes is one of our primary focus. And in the case of type 1 diabetes, if we look at metabolites associated with type 1 diabetes, there are several top molecules. I'm not going to try to pronounce this, but the abbreviation is LPC. It's a lipid which has altered levels in T1D, as well as, in this case, prior to the development of T1D. So maybe a biomarker of future development of T1D. It's also implicated in many processes that drive type 1 diabetes risk in a variety of ways, and therefore maybe a metabolite that is interesting to pursue further for involvement in type 1 diabetes. So those are just a couple examples. There's other traits that have similar association results that you can look up and gives you insight into metabolites that's summarizing data across the metabolite workbench. So we are interested in further improving these associations in a variety of ways. I'm not going to belabor this too much, but just to say that this is the first example of combining data from Workbench, and we're continuing to work with them to further enhance these results. For example, to change the types of analyses we do so that it provides effect size and direction, as opposed to just p-values, as well as maybe thinking about untargeted metabolite data, where the metabolite is a bit less clear, but it's a larger set of metabolites you could profile. And so thinking about how we can move beyond just targeted data, which is what we've used to date, where you know the metabolite, to thinking about how we can incorporate untargeted data sets in these analyses as well. So I'm just going to end then on thinking about how we can build on what we've done here beyond just the question of, are these metabolites associated with disease and levels of these metabolites. So one question we're quite interested in, given our expertise in genetics, is what disease-associated metabolites are also involved in causally in the risk of disease. And there are a series of resources we could consider using, some internal to Common Fund, some external, to address these questions. For example, QTLs for metabolites, so variants that affect metabolite levels. Are those variants also associated with disease risk? 2, what's the tissue cell type activity of these metabolites, using expression profiling, also inferring metabolite sensor pairs, which are analyses you can do from single cell data, for example, to understand the potential role in these tissues of metabolites that are disease associated. And then 3, what metabolites represent early biomarkers of a disease and phenotype, and this could utilize data at pre-disease states, some of which is in Metabolite Workbench, but may require slightly different analyses. Other resources in Common Fund, for example, A2CPS and others, are collecting metabolomics data in preceding stages of disease that could be used to address whether altered level of a metabolite is a biomarker of future development of a disease. So those are just some questions. I'm sure there are others, and yeah, it would be great, to get feedback from other types of questions that people would want to ask in this space, and combining data from either Common Fund resources and other external resources as well. But these are just some of our thoughts on what we could go from here. Okay, so that was fast. I'm sorry, but I wanted to make sure I stuck to time. And yeah, thanks very much. And yeah, like I said, we're continuing to think about how we could enhance the representation of these data and other data sets on the Knowledge Center, as well as what are the other types of analyses that could draw in multiple different data types that could be useful to users of the resource.
Beautiful. Thank you, Kyle. Sorry we didn't give you enough time.
No worries.
So with that, actually, I want to quickly share my screen and just invite you. Thank you for hanging in there a few more minutes longer, but we're just at the beginning. So next month, you're going to hear from our Data Resource Center. In addition to Avi, you're going to hear, Shankar, who you heard from today, you're going to hear from Avi Mayan directly and talk about the Data Resource Center. And I think this is really cool because it's a lovely complement to what we're trying to do in the Knowledge Center. And this is sort of an image that I generated when I was thinking about the DRC. It's sort of analogy to what I show with the child sort of walking through the ecosystem. I think the DRC has this sort of complex of a playbook. And the idea is that you can see all these different components of the Common Fund data ecosystem, and it provides you with all the data, all the assets in one place if you want to generate your own analysis. So different than the Knowledge Center, when we expose it to you already pre-cooked, a la the lazy man's lobster, with the DRC, you can actually dive in and do it yourself. And so you'll hear about that next month. And this is just a sneak peek of this really cool resource that Avi and his team have built that allows you to build your own sort of way of walking through the data ecosystem and build your own sort of playbook of what questions you might like to, scientific resources you'd like to access in the context of scientific questions. So we'll hear more about this in a month. And if you want to join our mailing list, please email this listserv, help@cfdknowledge.org. And it comes to like all of us. So that's great. So we respond really fast. The recording will be found from today on our YouTube channel, and I'll send the link around, but that's it. And also, if you just want to email me, Burtt, B-U-R-T-T, at broadinstitute.org, you can always ask us any questions. If you have any ideas for analyses, we really love to hear from you. And we're hoping that you tune in for the future webinar, which will happen in a month's time. Mackenzie, what date is it? It's escaping me right now. May 9th?
If you go back to Avi's slide,
Oh, it's there.
it has it there, but it's, I believe, May 30th.
May
31st, sorry.
31st. So it's after Memorial Day weekend, so hopefully everyone will be back. So I want to thank our speakers. This was really great. We packed in a lot to you today, but I appreciate you all hanging in there, and I appreciate all the speakers' time and careful sharing of really cool data. Thanks, everyone. Take care.
Thank you.
Thank you.
Bye.